Okay, hi everybody. That's much better. Yahoo! I think given what we had, I knew 5 minute break and let's just be one group, shall we? Okay. So I'm gonna wait for a second for Adam. We're waiting. I know. So we'd like to really say thank you. Thank you very much. And thank you to all our speakers for giving us such wonderful material to add to what we know and give us some inspiration for this coming conversation. So here I would like to say in the Google Doc, if you look at the way it's organized, and Google has this nice feature now up on the left there, where you can kind of see the headers of the sections. So these are the questions we kind of proposed to our speakers to take on in some way. And they weren't like required to take them all on. They take on the ones of their interest. And we asked them to also share, as they did, some of what their next step or next challenge ideas they all have. Another thing we asked them to think about was, and Cody mentioned this, I'm not sure who else, but the challenges that are both opportunities in the sense of, Cody, the one example you gave, so now the department and the medical school at the university is interested in your samples more than they have been before. So now they're collaborating with you. Yay, go team, more use of the specimens, more potential things that we can learn. At the same time, the regulatory system on a campus like the Department of Health and Safety is all of a sudden perhaps a lot more interested in what you're doing and how you're doing it, and maybe adding sort of regulatory permitting and other reporting processes to what you're trying to do, in addition to what y'all are already dealing with in terms of permits and collecting and things like that. So I personally, for example, am interested in process and thinking about, I mean, we all share it's very exciting to see the outcomes of a piece of research, but what can we say about what we need to do in the future and what we're learning, and what are both the opportunities and challenges involved, and what can we learn from what each of you has then experienced since COVID in this respect. Another one would be a very simple, concrete one. And for this, y'all, I can't resist. I use the CIA example of a go bag. So Nancy Simmons gave the example in our group when we started one of those in the CTAF conversation about the day came when she couldn't get into the AMNH anymore. No access. Well, the Mammals of the World book she needed, $800 tome, was sitting on the shelf in her office. No can do. So one of the first things she had to do, we had to buy another copy. Donut had to go dig up some money, go out, whatever, get it digitized. So there was a delay in that sort of, the information I need exists, but I can't get my hands on it. So are there kinds of things we need to be thinking about? I don't know what they are. Y'all will know better than me. What are we going to need to have handy if all of a sudden we can't get into the building? If we can't get into that freezer? If we can't get, how are we going to make sure we can get access or get some special arrangements? What do we do? And I don't know I only know that one example with Nancy, but you may have others. So thinking about those kinds of challenges. And that's sort of that new policies and procedures thinking about for next time. So in this document, and I'll see if I can scroll down effectively, or up rather. If you go down, you can see what we're looking for from you is sort of those 3 questions. Here we go. So what most resonated with you out of all the things that you heard? What was the thing that stuck out to you? Could in any shape or form, process, product, need. So what most resonated, and maybe a new idea that popped up in your head, or like a new idea could be, oh, I didn't think about this other need we have. It could be something we didn't talk about. And the last, so where do we go from here? Which you can see people have already been chiming in. So we had thought about perhaps doing this in 2 breakout groups and then coming back together, but I think we can do this group with this way. Yeah, I see people are starting to add to it already. And feel free, is there anybody that would like to share just out loud while we're typing in here what most resonated with you? And if you don't have access to this document, we'll try to capture your ideas. Yes? Um, I'm sorry, I don't know your name. Mike? Hi, Mike. I'm Debbie. Nice to meet you. Yes. Yeah, Andy's gonna let you so the people online can hear, and Cody's got access to my computer so he can actually see if people online, please get your names and get your ideas in there too. Please. Ah. Oh, there we go.
Sweet, thanks. So I'm the curator of mammals at the North Carolina Museum of Natural Sciences.
Ah.
I have probably 8 freezers full of backlogs of stuff that, I mean, we just took some seals out that were collected before 9/11. So, decades long backlogs. And so the reality is we have so many specimens that are frozen whole that just no one knows they exist. They're just, and every few years, I mean, I've only been there 4 years now, but when we defrost the freezers and inventory stuff and prioritize things, it seems almost like we should be cataloging these things it's almost like we should catalog them at that point when they're coming in, at least for these whole specimens that might be useful for colon samples, or because I can picture all of the bats that I have in the freezer right now, that if we had prepared them with our standard preparations, we didn't have colon tissues saved, for example, and so we have standard tissues that we save, the heart, liver, kidney, and muscle, but it seems like we should be potentially saving more things. And anyway, so it's just something to think about. That really resonated with me, is like expanding our purposeful preservation of different tissues, but also potentially, maybe it is important to save whole specimens frozen, especially with bats and rodents, because they don't take up a lot of space. So anyway, that was kind of cool, and I've kind of had a few instances of sort of serendipitous stuff where I have been lucky that we hadn't gotten to preparing specimens because they were still frozen whole, and it was like, oh, now we can do something with those that that it was just like accidental that we hadn't gotten to them yet. So I think those were important things for us to consider. And so the methodological approaches, I think their standardization and publishing protocols and stuff is what's important, because even like some of the stuff Kelly and Jocelyn and Molly and others have presented, it's like, I didn't know you could do a lot of that stuff, and I've kind of just assumed that everything that we're collecting is perfectly sufficient for whatever, microbiome or viral scanning and stuff, and maybe it's not. And so that was interesting.
Thank you for sharing that. I would add really quickly what you pointed out at the beginning is awareness that you have specimens that nobody else knows exist. Unless a researcher, for example, happens to know that they're sitting there because they were part of collecting and putting them there. I think you raise a point. There's a new standard coming for collections metadata, where if you sat down and you had the time, of course, you would capture that as a metadata record. You would publish that to somebody like GBIF and say, I have a collection of bats. This is the status. This is how before you digitize each individual specimen, you would have that metadata up there for discovery.
Freezing whole animals at -20 is cheap. At liquid nitrogen, not so much.
There you go. I think, yeah, I don't know.
Yeah, thanks, Mike. It's a great point. We're actually just starting an inventory system with a freezer number when stuff comes in, because we get requests all the time, like from exhibits. Do you have a frozen finnick fox we can taxidermy? We're like, we have no idea what's in the freezer. Can't even walk in there. So
We have no idea what's in the freezer.
yeah,
Yeah. No.
that was a surprise to our new director, who's an anthropologist. But
So that's where are they? Not only in nature, but where are they?
yeah, I think the main thing I want to point out too, and to your point, Mike, is being a person that receives requests, we get all kinds, and we talk about collaborating across the user community. We have to think outside traditional biodiversity data usage that we know is valuable to us, right? So, and that's happening, right, but beyond all the stuff we've done, but we can't predict every user community, but like preservation and these metadata are critical. I can't tell you the number of requests that we get where they say, I need kidneys that were not in DMSO and that were in EDTA, and we can't give them an answer. We can't even tell them how many kidney samples we have, right? And that's because they were collected for different purposes. They were collected for systematics, and it doesn't matter, right, to generate Site B. But now, as we're moving forward, and if we want to argue that pathobiologists are we make all these papers at museums with this rich repository, and then we do our analysis, and we're like, well, it's kind of a rich repository. It depends. If you're doing DNA viruses, great. If you're doing RNA viruses, no. Yep. So we've got to be honest with ourselves, but also increase our ability to track those data that are useful to other people. So then because they don't know, they don't open like even this project we did with Colleen, she could only use different words for tissues there to find out. There was no field for preservation type, tissue type in GBIF at this time. This was 2021.
Sure.
Right? And so it's like, how useful is TISS to a pathobiologist? It's not, right? So I think just thinking how we can expand our fields, and I know it's unlimited, you can't expand it for everyone, right? But at least for the pathobiology stuff, because we're arguing it's really valuable, and it is, but it's not if it's inaccessible to the people that need to use it.
Yeah.
Well, I would like to just add to that. So typically, we're thinking about, so I do on-the-ground wildlife disease surveillance work, and I do voucher specimens, but there's really been a movement away from lethal sampling, like collecting everything and taking non-lethal diagnostic samples, which you can still pull Site B and different biomarkers for species identification, but you can also do a plethora of different viral screening, different pathogen screenings. And is it common to have deposited swabs or diagnostic samples that are not tissues or organs, not necessarily linked to a voucher? Yeah.
I'll just give a brief example from the Field Museum. So to not knowing, we have a philosophy of not giving a catalog number to a tissue only. Right? And so if it doesn't have a voucher and it's a tissue only, we don't assign it a catalog number, which means no one knows about it outside the museum but us.
Yeah.
And so we're doing a project right now where we're not killing everything because we're doing capture, mark, recapture, but we collecting 10 samples per animal. And there's no way I'm not cataloging those 10 samples with a unified number. So the voucher is the tissue at that point, right? And so yeah, it's not going to make people happy, but it's like that we can't kill everything, right? And we probably shouldn't be in certain situations, but yeah, I think that's what we're doing we're starting to change it at the Field Museum, but it's like moving a boulder, right? Mm-hmm. So
yeah, I think Verity had her hand up, and this young woman in the back, the Verity had her hand up before that. Sorry,
I was adding this into the Google Docs. But going back to Mike's comment about having freezers and cataloging before you prep and everything like that, the only concern, and this is something that as an institution we probably have to face, is freezers die. My previous collection manager that I replaced would catalog things as they came in, and then there was a freezer meltdown, and all those specimens got lost. So you have to address that as a potential challenge. If you don't have your freezer on generator backup, do you have a system in place to maintain those in perpetuity, and what that looks like? And so that's a challenge that everybody has to, I think, try to figure out a way to find that good balance on. But just wanted to add that comment.
Yeah, no, she's next.
I'll be back.
Also adding on to the freezer discussion is that the tissues will still degrade at -20. Like the specimen within maybe a decade or two may not be as useful as when it was fresh. So unless it's like in liquid nitrogen, its usefulness is timed.
I want to reiterate in the Google Doc too, there are people, like Teresa, who's saying, wait, please catalog now. Don't wait. Yes.
Maybe it's just worth to invest in the science of preservatives at this point. I know I've been working with a company that's developed a new preservative that's great for keeping samples for microbiome analyses and tissue analyses for the DNA level stuff that lasts in perpetuity at room temperature, right? And so, but we need to maybe get away. Obviously, liquid nitrogen is great, but it's high cost, high carbon emit, that we'd have to put into it, a lot of energy, a lot of, things there. Do I think they're absolutely valuable? Yes, but maybe it's worth the chance. Maybe it's worth to invest and find chemists that would be able to work with us that could synthesize molecules that would allow this stuff to stay stable much, much longer at room temperature.
Mm.
So just a thought.
Cool liaison with spinach, Andy. That one. Yeah.
Yeah, I think just going off the same things, I'm trying to write something now that's like linking field procedures to preservatives and buffers that are actually useful in the lab. And I'm finding so much trouble like finding actual like protocols and papers that explain this buffer was really useful, this one didn't work as well, and things like that. Like I keep going back to the like DNA RNA shields like protocol, which might not even be, I don't know how accurate it is, but like sometimes that's the only time I can find that information. So I feel like connecting like molecular biologists, like Molly, you've been doing this, but like connecting molecular biologists to the field biologists who are actually collecting them. And Kendra, you're probably, most about this, maybe like, and how best to collect the samples that's actually going to be useful in the lab, I think is really important too. And I feel like there's a gap there, especially maybe for grad students who are just starting out or whatever, they're starting these projects up. Yeah.
For me, you point out a thing that I've noticed a lot in science is we're very interested in, like I said, in the results. And we often either don't have time or the space publishing to really talk about the process. We tried all these things and then we narrowed it down and this thing worked, so we did our whole experiment. But we don't have a lot of space in the paper to say, we tried this, we tried that, this worked, that didn't, let alone a coordinated way to share it, right? There's a resource in my brain, I can't remember it now, I'll look it up. But it was about how, like say you use DNase or RNA later, or what's the other one called? There's these different products, and you use one in your lab, and you use a different one in your lab, and so they actually came up with a system for assigning identifiers to those product names, so that when you refer to them in the literature, it's like the same problem with gene names, where you have to have this concordance between what you called it and what somebody else called it. And yeah, you need ways to find all the papers that have that. They found a way to do that with tools, protocols, and products like DNase and RNA later. If I can find that resource, I'll share it.
Yeah, there was a paper that came out of the NHM a year or two ago where somebody looked at all the different preservation techniques and compared them using different sample types. I can't remember who it was.
So one thing that I guess, and I think I put that first bullet up there while we were talking, is that we have lots of examples of pathogen research already engaging museum collections. But if you look at this group, outside of Rick, everybody has some affiliation with museum collections or has had an affiliation with museum collections in the past. And I think Rick's connection comes through me and some other folks that he's working with. And I think that actually kind of going back to, the 3 billion GBIF records that we all, we're very proud of, right? Having those data out there, but it doesn't get out to the community of scientists that really do the hard work in this space. And I think there is a opening for sort of reconfiguration of a data portal or a data aggregator or a GBIF for public health sort of portal that would allow people that are, say, at the Mayo Clinic or CDC or wherever they may be at to discover those samples for the first time, because they're not going to GBIF or iDigBio or wherever it may be, VertNet, to find out how many bat samples this field museum have, right?
Mm-hmm.
It's more happenstance that even if they do, even I know on our own campus, I'm sure Verity can attest to this and Pam can attest to this, that we have great medical schools that have, and schools of public health that have great scientists in them, but most of them don't even know exist across campus, right?
Yeah.
And I think if they knew about these sort of things, those connections would be much easier to make. So I think there's a definite need, at least from a data aggregator point of view, to maybe think outside of the typical biogeography, phylogenetics, systematics, and taxonomy group that could pull them in. I also think for us as museum scientists, I think I think Adam pointed out, we're built around systematics and taxonomy, right? We have systematic collections, but a lot of these labs that do this really high level research, it's not uncommon for them to write in sample cost and their NIH proposals.
Lots of money.
And we may need to start consider charging for sample access, I think, to be able to facilitate the science. And I think that's not something that we're not accustomed to. We work on a reciprocal sort of process, but the reality is people need a lot of samples.
Mm-hmm.
It takes a lot of time to pull those samples. It takes a lot of time to go to the field and do that work. And I think that in itself may be a mechanism not only to help prop up museum collections and the infrastructure that's needed, but also would help facilitate that community to seek out museums more readily for this type of research.
Yeah, sure.
I think Rick had his.
And I think also I want to push us to the next question if we've kind of gone through what resonated most. But if we haven't, just keep going.
I guess it's also, going back to your medical school and kind of what Neil was talking about, that kind of resonated with me a lot. And again, my general thing is we need more time, generally.
Mm-hmm.
But to be honest, if it wasn't for my section head being at Michigan, I would have never met Cody Thompson, right? And so I think a lot of the folks in kind of the pathobio virology thing, we're kind of, using like standard lab models. We're not thinking about wildlife as much. I guess CD was just this month released a thing for wildlife training. My campus prior to myself and Laurel Yo had never done this with wildlife. So again, explaining the value I think is quite important, and then also getting the grantors' head around it that it's far, I think my opinion after working with y'all is that it's far more real and what the real world is versus just looking at a rodent that has been captive and bred for 200 years, right? You get a more real world picture of zoonoses by looking at a bat or a rabbit or a rodent or a squirrel or whatever it is. Not so much of, I take a pure virus and I throw it on a thing and see what happens, right? And so I think what resonates with me is obviously, getting people connected, and then also trying to find, what Neil said, which is the same problem that Cody and I've had, is like, getting, the folks that are at NIH that are hardcore T cell people that work on, T cells, why bats, right? And getting their head away from the these kind of stuck models for working on mammals, for that matter, that there's way more diversity that we should be looking at as far as zoonoses and health and all these other things.
So we're talking about process at different levels, process and connections across disciplines, process inside a specific discipline, for sure. Are there any more thoughts that people are itching to share on what most resonated before we move on to new ideas?
Just a quick comment about
Go,
that.
Andy.
I mean, it is one of the ideals of the extended specimen network. You can envisage it as layers of an onion, and the last layer of that onion is extending out to other communities and trying to link our data to other sources of data in order to make those connections.
I think there's the Kading and Kingston paper that many of you know that talk about this need for cross-discipline work to answer big questions. And your time component that you bring up, Rick, is certainly tied into that, right? So this need for those things in a different vision and layers. So I think with that, please, if you have any more thoughts and we're missing them, get them in the doc. I'm trying to drive a Mac and get to there we go. Here we go. So new ideas and connections. We've kind of got a few of them around the same topic, but are there other new ideas that people are wondering about?
So I thought it was the Economist comment. I thought that was really interesting.
Thank you.
I thought that was like very, very profound. And that, if we had, and because they say these things like even, NSF, these kind of things, they say more transdisciplinary, multidisciplinary, and then we fall into these weird review sessions. But if you've just had an economist on your grant that said that this is not just because, scary viruses, that's what I usually use, but it has some sort of monetary value, and here's the data behind that. Maybe that is another angle to, potentially private funding, other approaches. And so I thought that was very profound.
Thanks. I'm glad I had the opportunity to share it.
Yeah.
Thank you. I have one, and Will, this is where I'm going to poke at you because I see you standing there over the wall, and I have no idea. Would you be willing to say something about, what is the potential for what you're doing or what LLMs might be able to do in any of this space? Did any thoughts come up?
If I stick a microphone in his face, he's going to have to talk.
Oh, there you go.
I think one intriguing thing is their ability to link disparate data sets together in a pretty fun way. Like we all know that you can get it to translate from like R into Python or something like that, but that ability I think is going to be really powerful going forward. Once we become able to probe latent spaces of those types of models, I think it'll be a really powerful tool to discover new links between things. And that could be really important for cross-disciplinary studies, I think.
So I started to, and again, I don't know enough. I'm just putting it out there and we can talk about it later. I was thinking about that, what you just said and what Rick was explaining, I think, or somebody you all, about the mathematical computational algorithms to sort of remove the host so that what's left is the RNA sequence, whatever. I understood that right. You're sort of doing this analysis to understand if you have a good baseline knowledge of the sequence of the host. So, and that's the kind of thing it seems like LLMs could be good at, yes?
Yeah, that's the embedded space. They link semantic things together. So theoretically, there's a world where it could interpret all of these like proteins, amino acids, things like that in a way that's more complex than we could tell it to do on its own. So that's certainly something that's going to be interesting to see in the next few years for sure.
Large language model, so ChatGPT. Okay. Other thoughts on new ideas and connections? We already talked about this connection between NIH and NSF and medical departments, and what else? New ideas, connections. Yes, Kendra.
So I work for a nonprofit. So that's another space where I think there's the opportunity to make connections. So we have a diversity of staff. So we have an economist who, what his primary job is to, like for the pandemic, the cost of prevention versus the cost of reactive measures. And that speaks a lot to, cost saving by doing disease surveillance work and getting ahead of it before it develops into a pandemic, which is much more costly. So there's that sense. Then I think for natural history collections, when discussing metadata and what you would like, to include in that metadata, I think having maybe webinars or materials so that people who, like myself, who go, I mean, just because I have a natural history background, I understand what is wanted, but my coworkers, like just getting them to voucher specimens, but also understanding how they should be collecting this additional metadata, it would be kind of a educational output for people who are doing on the ground type of work. So I think that's where the disconnect is if you're working with people who don't have a background in natural history collections and what type of metadata is required or requested, they're just they don't know what to do, so they don't do anything.
I would add, I think there are parallels to that in other disciplines. We have a group we had at iDig Bio called Integrating Collections and Ecological Research. And it was that same sort of, how can we make the data we collect as botanists, as bat researchers, more relevant to ecologists, whatever what they need, but how can the ecologist also voucher and make sure they collect what they need, but it's more useful for the systematic taxonomy community as well. And there were some very fruitful conversations that came out of that, but the same need there to understand relevance, what to do. They don't know how to voucher. They don't know where, whose door to knock on. So work to do in that. So thank you for that.
Well, I'll bring up the same comment that I brought up yesterday. It's a case of not only collecting the metadata, but databasing the metadata and then publishing the metadata and making sure that that is available. And I wonder whether we aren't at the point now where we need to publish our tissue collections as a completely separate resource from our voucher collections.
Oh.
Obviously linked together, so the tissue comes from a voucher, but publishing our tissue collections in a completely different paradigm so that you can publish all of that metadata and make it more available to the people who need to use it.
So I guess the other thing is I mentioned up in my talk is this idea of protein preservation and protease inhibitors and trying to be able to be ready for when we do have a protein sequencer available.
Yeah.
Anyway, so I think, thinking about proteins and being more protein centric, I think in the future might help us a lot. Versus getting, and maybe obviously it's great if we can continue to do liquid nitrogen. I'm not saying not do that, but also thinking about how to long-term preserve proteins, I think is really an approach, because then we could use antibodies and protein sequencing to solve a lot of these problems that are in the long term, basically.
So we took an opportunity here to remind you what's happening as we're having this conversation, right? It's cool, right? So, oh yeah. So are there more new ideas? I think we have new ideas, new connections, things we haven't brought up yet that we need to make sure we say or ask.
This isn't quite a new concept, but I think it could be picked up more. So as Rick's mentioning, you have tissues that are old and it's difficult to do traditional PCRs to pull out this kind of what the negative positives for different viruses or whatever you're interested in, but actually taking the lab to the field. So rather than worrying about cold chain, so I work right now in the rural Philippines, liquid nitrogen's not happening. Dry ice that's not going to happen for us. So we have to get around those issues that I think are going to be prominent in a lot of different countries. But there's been huge advances in being able to collect a sample from an individual, hold that individual in a bag, do entire PCR. I mean, they've even come up with PCR-free protocols, nanopore sequencing, and within 12 to 24 hours, what you have full genomes of viruses and the host. And then that can also inform, perhaps we should voucher this individual. because we're seeing multiple co-infections or a single infection, and would be great to have in and kind of guide vouchering specimens. Because unfortunately, I've had the luck of everything I voucher ended up not being infected with anything. So, skipping the step of putting it in liquid nitrogen, then having to worry about getting to a -80, backup generators because you've got power issues, where you can do all of this in the field at a BSL2 level, which, does limit what you can do with some of your samples, but it gets at a quick answer to what you're looking for.
That sounds so amazing.
But I think thinking bigger picture here, just because an animal is not infected doesn't mean that that animal is not valuable.
It would be good to know.
Used for all sorts of other things.
Sure. It could also
So-
be good to know at the same time you do that what the reference library is, because what if it's unique? What if it's undersampled? How many museums actually already have that specimen? Or if you could figure out a way to ask that. Yeah.
Yeah, I think. I suspect Andy's thinking other questions, but negative data is also good data, right?
Oh,
And I think that we oftentimes don't
Nope.
like that answer, but
Nope.
for example, I know this state of Michigan's rabies testing lab have well over 1,000 some animal submissions each year, and the percentage of positive animals is way less than 0.5%. But that's not the data that's reported, right? The 99.5% that are negative are not reported, and that's really it's too bad, and it's a little bit of a travesty. But certainly, especially thinking about this research area and the relevance for public health, I think that's important, because I think right now a lot of, at least what we're feeling on our campus, is that a lot of our folks in that space are a little bit petrified of everything, right? And it's not, and it's because we don't know. We don't know what that percentage of wildlife that is actually infected versus those that are not. And I think that if we don't know those numbers, we can't really give that information to the general public either. And that affects everything from people that want to go hunting to people that are just enjoy nature, right? So
You reminded me, and it comes back to what Kendra said too, about plant folks who study plant pathogens, and they keep a dual data set that they have to repeatedly sample, because they have to have a sample that's a control, right? They have to have this plant tissue sample doesn't have the pathogen, the rust virus or some other thing, and this does, and they have to repeatedly test that. And I was thinking of your sampling, where you would go and sample that bat, and it would be negative with your tests in the field from, I don't know, wing clipping. I'm not sure what you do for that. Thank you. Swabs. So, but you might recapture that bat in the future, and it might be positive. And then you'd have the relationship between those two events as well. So that, interesting. So, yeah, I don't want to stop you. Keep your thoughts. We do want to move on to the last question is, what do we do next? So if you're still sharing the sort of question we're on now, which is new ideas and connections, that's fine. We're going to wrap it up in about 5 minutes. So, Adam,
I have a question for Kendra.
Talk fast.
Yeah, question for you with like the nonprofit thing, and this come up like and like I feel bad, I thumbed down you and you said no vouchers, right? So changing this paradigm, and that was part of the thing y'all found in your bio paper, was people said, well, we reach out to collections, they don't want our stuff, right?
Yep.
But I'm curious, in your situation, this bugs me about Predict. Not only do they not collect any vouchers, but those samples are in a private person's lab that aren't accessible to the public.
Yep.
And one of the arguments we argue for big money and NSF and putting in this long term is that we make this material publicly available. We do it with some caveats because we're people, right? But in theory, that stuff should be available. How would it work? Like if you went to and said, okay, well, I'm going to collect 700, swabs from Taphozois and Oman, and you might not screen all of them, but you do, like, would be willing like to donate that? So like the question comes up like in permits, do you have to tell the people in Oman that okay, we're collecting this as EcoHealth Alliance, but we're going to donate the specimens to the Field Museum? Because then it also a whole other element of also like ownership. And so I'm just thinking like,
Nagoya.
yeah, thinking about we require significant documentation, but if you add in a museum, sometimes I'm just curious, like if that, like with your situation, giving vouchers to AMH might be easier, but have you ever tried to give like hundreds of samples of just rectal swabs to a museum and they've been like, no, or your bosses have been like, no, we don't want to do that?
So yeah, we follow Nagoya protocols like it's the Bible. So all of the countries I work in, they're more, I mean, we've had requests for tissues of all types. But yeah, we have most of our stuff stored in 2 separate biorepositories because museums typically want the whole voucher along with those swabs. But yes, now I have 24,000 swabs sitting in freezers that I don't have a voucher to match with that. And so our partners, sometimes we send stuff to like Rocky Mountain Laboratories for testing, but we have data sharing agreements across the board, export, import permits with every documentation. And that might be my museum background that has trained me to do all those things. But yeah, no, I mean, we are very cooperative with donating, but a lot of museums want that voucher with it. And when we do supply vouchers, we give them everything that we can. It's just this, that's kind of what I was asking, if would you ever be willing to take diagnostic samples that are not lethally collected? Because we get quite a bad name as virus hunters, and there are some groups from different countries that wipe out entire populations just to collect a liver sample, and then they burn the rest of the animal.
Okay.
And so there's been a big push to move away from that and be more non-lethal in our sampling. But sometimes we hold on to the samples in the hopes that we can get funding to screen them for additional pathogens. But if not, yeah, we want to find them a permanent home. We never want to throw anything away. So, yeah. And I mean, I agree there are faults with the Predict project, so you're not going to get an argument out of me for that. But I mean, that's the case where they just didn't have the guidance of working with museums to understand the importance of vouchering specimens. And I was not part of that project, or otherwise I would have got onto them.
Thanks, Kendra.
Thanks. Yeah. Adam kind of asked my question, but I'm going to follow up, I guess, a little bit with that. Like in terms of thinking about swabs, for example, as vouchers, like can we accept that and move towards that, like collectively? Do we need a tissue sample with that? And can, for example, microbiomes be characterized enough to serve as a voucher that you can characterize the microbiome and identify it to the species that it came from? And I think in some cases you can. In many cases you can. And so is that a reliable voucher for a species? And like, I think a lot of people are thinking no, but there is this like kind of gray area where it's like, well, how are we supposed to preserve these things? And I think it goes back to Kelly's talk and how they're creating a different database for that stuff, but linking it all back together to the original animals. I think the complicated thing for example, your bats is like, are you tagging the bats and so you could still identify individuals through time, or are you, getting these swabs and maybe not being able to? Because my concern in a lot of these instances are like animals being, vouched, like especially when you're not taking the whole animal or something, that animals are being vouchered multiple times across places, or different parts are going here or there or whatever. And I think that's where it gets real complicated with not having the skull or skin or something like that. So I think that's something we all have to collectively decide together and, you know, put it out there what the protocol is and what is or isn't acceptable. And I have a lot of the, you know, old school folks will say, no way, like we can't be doing that. You should create your own repository, and maybe that's the answer, is you publish all of those microbiome swabs as an ecology data paper or something like that. But anyway, thanks.
So that's a nice segue. We really have to wrap up, but if you're willing to do this for like the next maybe 5 minutes, maybe it might take 10. We kind of did a little bit of this, but now the last question on that document is what next? What do we do next? And these, we don't have time to rank them. We don't have time to look for dependencies between them. We just have time to collect them. So feel free to share. What are your ideas for next steps?
Yeah, so going back to your microbiome thing, so we're in the process at Charlotte of developing bioreactors to actually take a small amount of a stool sample or any tissue that has microbes in it and culture that up, and then cryopreserve that in perpetuity. So we have model microbiomes from various organisms. We can do that from swabs. Yeah, absolutely. And we think we can do it from, we think we've done it, a friend of mine has done it from fish and other things as well, because we're losing these as the climate is changing, we're losing these microbiomes, not only in animals, but also in people. So having that as a potential voucher, I would be more than happy to help write a paper on such a thing, and potentially write a grant on something like that. But I think that's a great idea, actually.
Other next step ideas?
I think there's also a potential middle ground here. Why not use images as voucher? Why not take an image of the specimen? If all you need the voucher for is to confirm identification, you can identify things from an image, especially mammals. Fishes, it's a little more difficult, but.
Say that, Verity, in the micro. Say it so the online people can hear. Verity.
I would argue that mammals not always easy to identify just by picture either, especially a lot of rodents.
We're wrapping it up in 7 minutes, Gil, just so you know. Adam's got a next steps idea.
Just following up on Andy's comment, like there's a middle ground, right? Like I think there's always this resistance, so we need to think about how we don't lose what we are as a core, which is specimen based and voucher based, but also like not being so resistant to novel data sets that, to Mike's point, like one of the things we're struggling with our cataloging the material is we are capturing known individuals over time. So we have a longitudinal data set, which tells us an amazing thing. So this ecological data set, right?
Yeah.
And like at the museum, we're working now because we can't collect elephants and big ungulates, but we use camera traps. So we have that. At least we have the voucher point, space, and time. So thinking creatively without losing our core that we are voucher physical based, right? And just, yeah, I just think it's smart. Like you're exploring the tissue option as something different, right? Then it's like, well, then, okay, now it's not the voucher, but it's still something. But yeah.
In my brain, that's a growth mindset.
Yeah. I mean, in ichthyology, we do it all the time. We use photo vouchers if we collect something from a fish market, and they won't let us take the voucher.
We've got 2 minutes.
What interests me most as a collection manager is how to link all of this data, all the pathogen and the microbiome data together with the voucher specimen. Like, how are we going to reflect that in our collection databases, and how are we going to push it out to data aggregators?
Yep. My brain goes in that direction as well. Support for making that happen. Other next steps, ideas. What should we tackle next, do next, talk about? There you go. Okay, yeah.
I think, there's the bioscience call for One Health, and this always blows me away because we could never get money to do rabies work, for example, which has a human health component, but they would not fund surveillance or dog eradication because it's not directly applied to human, even though 99.9% of the cases are from domestic dogs, right?
Sure.
So
yeah, that was my point about domestic transfer as well.
Right.
Yeah.
So how do we tap into the human NIH funding, which some of y'all are doing by cross collaborating, but I think as a museum institution, right, IMLS isn't going to support us all the way for this kind of stuff, right? So figuring that out.
DOD has lots of money.
There you go. And we had a DOD, there's a U.S. Army person here. I'm trying to think of her name. Ooh, she does database stuff for them. I think, are we final ideas? If you have more, you can put them in the document if more pop up. And I want to say an amazing thank you on behalf of all of us, on behalf of Pam and Cody and Courtney and all of you who presented. Thank you very much. And know that Courtney's now, those are sketches that she's. You want to say something, Courtney? You want to say? Yeah, so, and she will be sharing them with you too so that you can give her feedback. So we want to reflect what you meant to say and how you meant to be represented. So there will be time. You can, yeah, jump in, Courtney.
Super, thank you. Yes, so these are rough. You can see they're pretty quickly scrawled up there. These will be polished over the coming days. I'll integrate more visuals, but I will work with Debbie and the team to share these out to you. That's one of the beautiful things about this kind of work, especially in digital form, is it's utterly editable always. So I really will value your eyes to this. If you see things from those of you who are presenters from your talks that are a little bit mischaracterized or could be better clarified in case this is shared out to a broader audience, please, I will invite you to let me know, and I'm happy to do that. Of course, this is amazing, rich content that you've all presented today. I'm sort of washed over with all of it in awe, and have done my best to distill this with the idea of audiences that could span both other experts in your disciplines, the people you're wanting to connect with, but perhaps also the general public. So that's something that I've been striving to do as we get into this. So this is not going to carry the depth to substitute for the talks that you gave, of course, but is meant as a tool, like a springboard for engagement and a conversation starter. So I look forward to your feedback to keep making these more relevant to all of you moving forward.
Yes. Was it Andy, wait, yeah, go. Repeat the question, please.
Okay, what are the biggest challenges for me doing like the work that I am here today? I think diving into a new discipline myself that I have a more surface level knowledge about, I've done some research ahead of time, trying to kind of soak up what I can leading into today, but being able to depict, I think, especially abstract concepts with a lot of interconnections. I mean, there's a real value in doing that visually. There's a lot of potential. Sometimes just distilling it into that nugget is the hardest part because I want to convey all of this. I want to I guess pass along as much of the complexity in all of these words and data that you're talking about, but there's obviously kind of that sweet spot to hit. And so I think that's always the challenge.
So again, thank you very much. And thank you to IDIC Bio and Vaughn. Thank you for helping tie all this together. So looking forward to seeing many of you at ASM 2024, and we're going to essentially reinvent this, but we have even a sort of extended time to do more of this and maybe a little bit of writing as well, and thinking about more about how to maybe take some of those next steps forward. If you have other colleagues that you know would like to be involved, that could contribute, I think of Fio Cruz in Brazil. I think those people would have some incredible insights for us on some of these topics. Please let us know. And thank you very much. Oh, yeah. Anybody up for a photo? You up for a quick photo? It'd be great. Yep. Cool. Sure. And to everybody online, we can't thank you enough for sticking with us and for contributing your ideas in the Google Doc. Thank you for sharing what's inside your head for all these conversations. Thank you. Ah, Neil, we're glad you're up for a photo. Yeah, shall we come up here?
I actually just saw another friend who's stranded somewhere else.